We are currently recording. I'm going to do a quick, very quick 5-minute or so review of how a neuron generates and propagates an action potential. So I'm going to quickly go right to a whiteboard and go through this whole story again with you. It's just a good, quick review. So this is a multipolar motor neuron that I'm drawing. We're not going to myelinate the axon. Here's the soma, the cell body with the nucleus. Here's some dendrites around the soma area. And we know at resting membrane potential, there's a high concentration of sodium ions outside this membrane and a high concentration of potassium ions inside. And like I said yesterday, that goes all the way around this cell, all the way around the edge. That creates a net charge of positive out and negative in. So at resting membrane potential, the cell looks like this all the way around the plasma membrane of this cell. If this cell, this neuron that's at rest, gets stimulated by another nerve cell, for example, so I'm going to put its axon terminals here, put one down here on the cell body. When this other neuron sends a signal, releases its neurotransmitter from its axon terminal onto the membrane, let's say, of this, here's the synaptic cleft of this cell body of our resting neuron, that causes a short-lived, localized depolarization of the membrane. So if the point of stimulation is here, we get a graded potential, a flipping of this charge, a mild depolarization around the membrane of the neuron cell body. Now, if this cell stimulates, releases enough neurotransmitter in a fast enough rate that these graded potentials, these waves of depolarization, are large enough that it makes its way further around the soma cell body, the waves of depolarization. And if this stimulus is strong enough, it reaches this cell's threshold. So if these waves of graded potential depolarization are strong enough, it reaches this neuron's threshold, and at the axon hillock here, right here, we get a massive gate opening, sodium gate opening, and sodium ions gush into the cell, much more dramatically than in a graded potential out here. So this huge channel opens up, sodium gushes in, and that's the start of the action potential, which is just depolarization of this axon. Once this opens, we can't turn this process off. It's an all or none. Once threshold is met and this gate channel opens, it's an all or none response. It's going to fire all the way down the axon. What that means is then in sequence, neighboring sodium gates open one after the other all the way down the axon, and sodium ions rush into the cell, causing depolarization. Think of it as a flipping of this initial charge of positive out, negative in. Because sodium gushes in, it ends up causing a positive net to negative net. So in essence, we're flipping the resting charge all the way down. And then as gates are opening downstream, sodium gates, the sodium gates upstream are closing one after the other in a linear fashion. Once these sodium gates start to close upstream, potassium gates then open up. I'll use a different color for potassium gates. I forget what I used yesterday, but let's just go to yellow. So then potassium gates open up in a linear fashion. Now we've started the act of repolarizing the cell, because when potassium gates channels open up, potassium rushes out of the cell. And enough potassium rushes out that it repolarizes the membrane, meaning we go back to a positive environment out here relative to a negative environment in here. That is called repolarization. And remember, during repolarization, there are things called refractory periods. Well, absolute refractory period actually is during depolarization. This cell, this neuron, will not respond to another stimulus during depolarization, no matter the strength of the second stimulus. However, relative refractory period is defined by this neuron will respond to a second stimulus during repolarization if the second stimulus is greater than the first one out here, the first stimulus out here that caused the initial depolarization. So that explains resting membrane potential, the act of reaching threshold and depolarization, the firing of an action potential, the act of repolarizing the cell. Just to remind you that oftentimes so much potassium rushes out during repolarization that it overshoots the initial relationship of positive to negative, and it makes the outside even more positive because so much potassium leaks out. It hyperpolarizes that membrane. So when you see the term hyperpolarized, just think so much potassium leaks out during repolarization that it overshoots the initial out here, the initial resting positive to negative relationship. Okay, if you have any questions, you're free to ask me either right now, interrupt me, or at the end of this bit, I can re-explain anything you want. But we need to get on to the final material for tomorrow's quiz, and so I'm going to quickly do that. First topic is we're going to talk about synapses. So you know what the synapse is. I'm just going to redefine it for you. It's basically the junction between a neuron and its postsynaptic cell. So a synapse can be neuron to neuron, where we have a presynaptic neuron and a postsynaptic neuron. A synapse can also be from a nerve cell, a neuron, to an effector cell, like a muscle or a gland. So both of those junctions, these communication places, are called synapses. Now there are two major types of synapses. I think your book mentions the other more rare ones. I'm mostly concerned for our purposes in this basic class that you just know that there are two major classes, and those are axodendritic synapses and axosomatic synapses. Now that you know this language, it's pretty easy to decide or determine what those mean. This means going from an axon of a presynaptic neuron to the dendrites of the postsynaptic neuron, or going from the axon of the presynaptic neuron to the soma or cell body of the postsynaptic neuron. I'm going to tell you, they mostly have the same effect at the postsynaptic cell, but it's just where the axon terminals are placed, either on the dendrites of that resting cell, postsynaptic cell, or the soma, the cell body. Neurons can have, this is kind of mind-boggling, but our nerve cells, our neurons can have up to 10,000 axon terminals at the end. So when I draw my picture of a typical motor neuron, I just usually draw 5 or 6 axon terminals. That's a gross understatement, simplification. Our nerves can have 10,000 of these at the end, which is mind-boggling. And they can also receive that many at their dendrites and soma as well. So the number of stimulations that a neuron can receive and transmit gets to be, number-wise, gets to be sort of mind-boggling. Now I'm going to go into types of synapses, so I'm going to layer over this. We're still talking about synapses, and these are not classifications of synapses like axodendritic, axosomatic. These are going to be classified into chemical and electrical. So now we're examining what actually occurs at the synapse itself. I'm going to go over, I typically lecture about electrical first, but because your textbook talks about chemical synapses first, I'll do that just to be consistent with your book. We know mostly about chemical synapses already, meaning us in this class, because that's what the neuromuscular junction is, a type of chemical synapse. So I'm going to say a few things. First, these types of synapses always release neurotransmitter because that is the chemical that makes them chemical synapses. There is no physical connection between cells, and that is provided for by a synaptic cleft. So that word cleft just means that's the space, and it's not airspace, by the way, it's a fluid-filled space between one cell and another. So it's worth noting that there's no physical connection whatsoever. This neurotransmitter is stored in vesicles. We know that from our neuromuscular junction. So at the axon terminal, the neurotransmitter that is manufactured by the neuron gets stored in those bubble-like structures called vesicles. This is, I'm going to put a star, an asterisk. In this type of synapse, communication is always unidirectional. That means we always go from communication, always goes from the cell releasing the neurotransmitter to the cell that's receiving the neurotransmitter. That sounds basic and intuitive, but the electrical synapses don't always behave that way. That's why I made a big deal out of that. So now I'm going to talk about, your book lists steps of neurotransmitter release. It's really basic, and we already know much of this discussion. First is, I'll write that out. I do want you to review these steps in your book prior to the quiz, but they're fairly simple. So I'm just going to write steps to neurotransmitter release. Okay. Number one is just the action potential arrives at the axon terminal. So you know how an action potential is generated in a neuron, and then it travels down the axon. So that action potential, that electrical impulse, aka depolarization of the membrane, arrives at the axon terminal. 2, voltage-gated calcium channels open, and calcium, it's called influxing. It enters the axon terminal. So calcium from outside of the nerve cell then enters that axon terminal. Step 3, I'm going to talk a lot about now. Step 3 is just titled Calcium Entry Causes Synaptic Vesicles to Release Neurotransmitter by Exocytosis. So I'm going to write neurotransmitter release by exocytosis. And I'm going to expand on it. Your book says these things, but in my opinion, it doesn't explain them all that well. So I'm going to do that in a picture format. I'm just going to go to the next page. I'll save this in case you want it or need it. So we have, here's what's going on. Here's an axon terminal. Let's just say this is a postsynaptic cell down here. It can be a muscle, can be a gland, can be another nerve cell. Doesn't matter in this case. Step 1, the depolarization, the action potential comes down the axon and it arrives at this axonal terminal. Voltage-gated channels on this membrane open up from this electrical impulse. They open up, and calcium ions out here, outside of the cell, enter this nerve cell. Now we know that neurotransmitter is stored in vesicles in this terminal. So the neurotransmitter are just these little dots. The vesicle is the bubble that's containing them. When calcium enters, it binds to a surface protein on this vesicle. So I'm going to put a surface protein out here, and a calcium ion binds to it. That surface protein, that letter Y that I've drawn, it's got a fancy name, but it's a simple concept. It's called a synaptotagmin. That's just a surface protein on the vesicle itself. Calcium binds to this synaptotagmin right here. And your book doesn't say this, and so I'm not going to test you on this part, but that then causes this protein to change shape a little bit, and we then call it, it's then called something, it's called a V-snare, but just know this, calcium binds to this synaptotagmin on the surface, and then this vesicle migrates to the edge of the cell, and that synaptotagmin with calcium bound to it that changed shape then binds to something on the surface here called a snare protein. So a snare protein looks like this. And it binds with that calcium-bound synaptotagmin. That causes the vesicle to fuse with the membrane. So I'm going to draw a vesicle right here fusing with the membrane, and it releases its neurotransmitter by exocytosis. That whole process is called neurotransmitter release. That's step 3. So I'm going to just go back to this page. We just described neurotransmitter release by exocytosis. This neurotransmitter release by exocytosis is the rate-limiting step, what is called synaptic delay. Synaptic delay is just the time takes for neurotransmitter to be released from a presynaptic neuron. And this takes the longest time in the communication transmission from one nerve cell to another cell. So when I was in your seat, we were asked, let me go back to this page. Sorry. When I was in your seat as a student, we were asked, what do you think takes the most time for this whole communication site to happen? Most students, including myself, thought, oh, the longest step has to be for this neurotransmitter to diffuse across this synaptic cleft and actually bind to receptors on this postsynaptic cell. That's not the case. The rate-limiting step is synaptic delay. This process right here is what takes the longest in neurotransmitter communication and chemical synapse communication. It's the release of neurotransmitter. So then step 4 is just the neurotransmitter. So I'll go back to here. Step 4 is that the neurotransmitter diffuses across the synaptic cleft. That's supposed to be. I'm trying to write quickly. It's not working. And number 5 is the binding of the neurotransmitter on the postsynaptic cell. Obviously, it causes something to happen at that postsynaptic cell, and typically it opens ion channels. Opens ion channels, and that's what causes a graded potential. At the postsynaptic cell. Okay, so those are the 5 steps of that business. We talked about synaptic delay. Now I'm going to briefly talk about electrical synapses. And your book doesn't make a big deal about this either. So let's go back to, now I've got 2 new pages. Let's fill in this one. Electrical synapses are different from chemical. Electrical synapses are much more rare. They're not super rare. They're mostly found in the central nervous system, mostly the brain, but they're not as common in the human body as chemical ones. And that's supposed to be UP. They're made up of gap junctions between cells. I know that you guys, most of you probably haven't taken Bio 1. It's not a prerequisite for this course. So I don't have time to explain the whole intricacies of what a gap junction is, but I'm just going to write it's a physical connection. So unlike chemical synapses, where there is no physical connection, there is a physical connection from cell to cell. And what this physical connection of this gap junction looks like, here's one cell, here's another cell. There are protein channels like this. And these proteins making up these gap junctions have a specific name. These proteins are called connexons. It's C-O-N-N-E-X-O-N. These connexons allow for the flow of electrical activity, the flow of ions. So these are electrically coupled physically touching and electrically coupled by these connexons to allow for ion flow from cell to cell. So here's some special considerations. First, and this is unique from the chemical ones, communication can go either direction. So I'm going to write communication can be bidirectional. Remember, in chemical synapses, it was always unidirectional. So flow can go in this direction or this direction. And the communication or the transmission is extremely fast, much faster than a chemical synapse. So I'm going to just write very fast transmission. These types of synapses are found largely in the brain. And what this does is this allows for our brain activity to have very coordinated synchronous activity. It's not always a one-way street or one-way transmission. Information can move from this cell and back to this cell almost at the same time. So we get a much more synchronous type of communication in our brain, which is good. All right, so we've done chemical and electrical. Now I'm going to talk about postsynaptic potentials. This is kind of complicated. I'll do my best to simplify it for you. So let's call up this other blank board and label this postsynaptic potentials. Let's decipher this first. We're just trying to find out what happens at the postsynaptic cell, the cell that's receiving the transmission from the presynaptic cell, and what happens when it receives that transmission. So that's what this topic is about. There are two major types of postsynaptic potentials. And we label those two types either excitatory postsynaptic potentials and I'm not going to write the second one yet. They're inhibitory, but we abbreviate this EPSP. That stands for excitatory postsynaptic potentials. So this is, remember, we're talking about what happens at the cell that's receiving the neurotransmitter, okay? First and foremost, during excitatory postsynaptic potentials, the neurotransmitter that is received depolarizes the membrane of the postsynaptic cell. So if you remember one thing about EPSPs, think the word depolarization. It depolarizes, I'm going to say, the postsynaptic membrane. Remember, the act of depolarization is an action potential. So it's causing something excitatory to happen. That's why we should equate the word depolarization or depolarizes with exciting or making something happen, like firing some sort of new electrical impulse. So we know the next bit. Sodium and potassium do their thing. I'm just going to write diffuse in and out of the postsynaptic membrane. These are called chemically gated ion channels, Why are they called chemically gated ion channels? Because it's a chemical, this, that are making these channels open up. So they're chemically regulated or chemically gated. So we know that we get a net depolarization. I'm going to repeat that. With excitatory postsynaptic potentials, there's a depolarizing activity. Instead of an action potential being generated on the postsynaptic cell, actually we get a graded potential. So I'm just going to write first. I'm just trying to erase that. First, we get a graded potential, and then if that graded potential reaches threshold, that cell will fire an action potential. So this term, excitatory postsynaptic potential, is really just describing in one term what we already know. So all of the examples I've given you up to this moment have been examples of excitatory postsynaptic potentials. The function of an excitatory postsynaptic potential is to help trigger, this is the goal, it's to help trigger the action potential at the axon hillock of the postsynaptic cell. So all of the examples that we've talked about in firing of an action potential on that receiving cell has been describing an EPSP, okay? The second type, so I'm going to layer over this. The second type is an inhibitory, so I'll write a number 2. It's similar to excitatory, but it has some major differences. These are abbreviated IPSPs. You can guess from the name what happens is when, for example, a neurotransmitter is released, it doesn't cause depolarization, it's inhibitory. So this type inhibit postsynaptic cells membrane. So we're not going to get depolarization. So that's first and foremost. So, and this action inhibits the postsynaptic cell's ability to generate an action potential. Okay. Most IPSPs work by hyperpolarizing the postsynaptic cell's membrane, not depolarizing. So most work by hyperpolarizing the postsynaptic cell's membrane. What this does this takes the membrane further away from threshold. So unlike a graded potential in the last example that gets a receiving cell's gets closer and closer to threshold, this gets further away from threshold, making it harder for it to fire or more difficult to generate an action potential. So I'm going to draw a quick picture and just try to explain this as best I can. Here's our postsynaptic cell, as always, this motor neuron. You know the story of an excitatory postsynaptic potential. I'm not going to go through that again. Here's the cell. It's still at rest, looks the same. Sodium out, potassium in, positive net out, negative in, all of that's the same. Rather than when this transmitting cell sends a signal, rather than causing a graded potential that depolarizes this receiving cell's membrane, it's going to hyperpolarize this cell's membrane, meaning making this a stronger positive and this a stronger negative. That keeps this cell from ever reaching its threshold. We're getting further and further away from it. So it's dampening this cell's effect to fire an AP. Think of it this way. In an excitatory postsynaptic cell, postsynaptic potential, if your electric circuit from the switch on your wall that turns on your light, here's a light bulb. These two are the same. If you walk up to that light switch and you turn that switch in an upward fashion, and you turn it up really slowly, you are mimicking a graded potential onto this receiving cell. So you're doing something, you're slightly depolarized, you're causing some activity here, but you haven't reached this cell's threshold. The minute you turn that switch up slowly and it hits the point where it fires an electrical impulse and turns this light bulb on, you've reached this cell's threshold and it fires electricity and it turns this light on. That's the same as reaching threshold, firing an action potential, and releasing neurotransmitter. And just like turning a light on in your house, once you meet that point of when you slowly turn this switch up, once you meet that exact point that causes this thing to fire and turn the light on, it's either on or off. It's not on a dimmer switch. So that's what an EPSP basically does. Excitatory postsynaptic potential. Now let's say you walk up to this light switch, same light switch that goes to a light bulb. And if you turn this switch not up to turn it on, but if you turn it further down in the opposite direction, you're mimicking an inhibitory postsynaptic potential. You're making this mechanism, this neuron, go further away from, not closer to reaching its threshold to turn it on, but you're going further away from threshold. And so this light will get further away. It'll make it more difficult for this light to turn on. That's sort of an analogy that I give in class to my students of the difference between these postsynaptic potentials. This all postsynaptic potential business, whether excitatory, inhibitory, we're describing what happens here at the light switch, here at the cell's receiving end. All right. Hopefully that makes some sense to you. Now I'm going to briefly talk about summation of neurons and then neurotransmitter effects. Then I'll give you a chance to ask questions, okay? So I'm just going to mention these two terms and I'll describe them. You already know from our muscle unit It's very similar terms and concepts. I'm writing spatial summation and temporal summation. You already know what these terms mean, or they should at least look familiar to you. I don't expect you to remember every single thing from the last unit, but spatial summation is when a postsynaptic cell is stimulated simultaneously by many presynaptic neurons. So spatial summation, if we have our postsynaptic cell here, and we stimulate it by one neuron at the same time, 2 neurons, 3 neurons, 4 neurons. The additive stimulation by these presynaptic neurons causes this postsynaptic cell to reach threshold more quickly. They summate. That's where the word summation comes from. And spatial means we're adding more and more the number of presynaptic neurons so that their effects summate or become additive. So this cell reaches threshold more quickly and fires its action potential. Temporal summation is just like in the neuromuscular junction business. Temporal summation refers to if we have a postsynaptic neuron and let's just use a simple example. We stimulate it with one neuron and it fires an impulse, releases neurotransmitter, fires an impulse, releases neurotransmitter. Now in temporal summation, we increase the firing rate from the presynaptic cell. So this firing, we just increase its rate so it gets faster and faster. It goes from fire to fire much faster. That increasing rate also causes this cell to reach threshold more quickly and then fire a response. Those are what those two terms mean. We know now that neurotransmitter effects from our last discussion that we just did. We know that the effects of neurotransmitters can be either excitatory and or inhibitory. So neurotransmitters can be excitatory, causing depolarization, or inhibitory, causing hyperpolarization, or both. So I'm just going to say can be excitatory. Remember that means depolarization, or inhibitory. That is hyperpolarization at the postsynaptic cell's membrane. And then I'm going to put a little asterisk here. Some neurotransmitters can be both excitatory and inhibitory. Here's a classic example. Acetylcholine, that familiar transmitter that we learned back in the neuromuscular junction. When acetylcholine is released on skeletal muscle cells, it's excitatory. It causes depolarization, okay? But when the same neurotransmitter is released on cardiac muscle in the heart, that same neurotransmitter is inhibitory. So when acetylcholine is released on our skeletal muscles, it causes them to contract and contract more quickly if more acetylcholine is released. the same neurotransmitter, the exact same chemical, when it's released on cardiac muscle, it's inhibitory and it causes less contraction. It causes the heart to actually slow down. It's not excitatory, it's inhibitory. That's really important to remember. What that tells us is the effects, that's supposed to be an N, the effects of neurotransmitter this is really important, depend on the type of receptor at the postsynaptic cell, not on the neurotransmitter itself. So the chemical itself does not dictate the effect at the postsynaptic cell. It's the receptors at the cell that dictates the response of that chemical. All right. Now I'm just going to briefly mention two different neurotransmitter receptor actions, not the types, but I'm gonna write neurotransmitter pathways. I've just tried to create a new whiteboard and it says I'm out of whiteboard. I only get 12, so I need to erase some of these older ones. So let me do this. This is the last topic, I promise. I'll call up a new whiteboard. I'm just going to write neurotransmitter receptors. What we're talking about in this last topic is the actual proteins on the membranes of the postsynaptic cell and how they're configured. And there are two major types of proteins and how they act. So the first one, the first type are called channel-linked receptors. Now these receptors are also called ligand-gated ion channels. So this is just language, and I get it. Believe me, I understand this is confusing. When you read this word, the term ligand, just think of that as a protein, because it is. It's a protein that transmits some sort of a signal. Okay? So it's just a fancy word for a fairly simple concept. So that word ligand is just think of it as a protein that's going to do something. So what happens is the protein receptor, the ligand, it changes shape, okay, when the chemical binds to it and allows things to pass through. So I'm just going to give you an example. If this is the postsynaptic cell's membrane, here's the ligand on the surface. When the chemical, the neurotransmitter, is released from the presynaptic cell and the chemical binds to this protein, this ligand, it changes shape and allows for things like, for example, sodium ions to go through. So originally the ligand or the protein, let's say, was kind of closed. After the neurotransmitter binds to it, it opens up and allows things to go through. That's called a channel-linked receptor. This type always receives neurotransmitter. So they're only involved in chemically gated or chemical synapses, and they always receive neurotransmitter, but they can be either excitatory or inhibitory. Okay, so those are channel linked receptors. Now I'm going to briefly explain G protein linked receptors, which are even more complicated, but I'm going to give you the simple Reader's Digest version, and you're not going to have to know any more than what I described to you, okay? So the second type, number 2, oops, or number 1, that's no good. So number 2, these are called G protein linked receptors. Here's the simple version. These receptor actions are indirect. First, I'm going to say this. These are much slower actions. And we find a lot of these types of receptors in the brain. They're also called, this should be in your book, metabotropic fancy word for not that difficult of an idea, receptors. The only reason is because they cause a change in metabolism at the receiving cell. Metabolism is just a word that means chemical reaction. So these are the same things. G protein linked and metabotropic mean the same thing. Metabotropic is just a little more descriptive because this is the end result, is usually increasing or changing the metabolism of the postsynaptic cell. These are typically used in what we call learning processes of the brain. Just like when you're trying to learn a lot of complicated A&P material, you're using a lot of these pathways in the brain. And they're slow. They're not super speedy. Here's what happens. G proteins are typically linked to a second messenger inside of the cell, which activates some sort of chemical kinase inside the cell or protein synthesis. So rather than this ligand protein just sitting on the surface, what we have is some sort of surface receptor, just like the protein channel receptors in the first example. Let's say there's a chemical that's going to bind to this receptor. This is more indirect. So this receptor doesn't just open up. What happens is it's physically linked to another protein inside of the cell called a G protein. And when this chemical binds to the receptor that is linked up with the G protein, that causes a series, a cascade of actions to occur. And it's called a second messenger system. What happens is this whole long process of converting one thing to another, for example, cyclic AMP might be called, or calcium influx might be used as a second messenger, calcium or CAMP. These are examples of second messengers that eventually cause something like protein synthesis. Long chemical processes is the point here, or That's supposed to be an I, K-I-N-A-S-E-S, kinases, to regulate or change the cell's metabolism. So that's, I know it's complicated. Here's what I want you to remember. G protein linked receptors mean that this thing is linked up to the surface receptor. It's indirect. It takes a long time for this to occur, slower acting. They have another name called metabotropic because oftentimes there's a change in metabolism of the postsynaptic cell, and that these are used in learning processes in the brain. Okay, I'm going to call it good there, guys. So let me just cut off my recording and I'll give you a chance to ask any questions you want.